Let's talk about ANP and BNP, right?
Okay.
Atrial natriuretic peptide, right? And brain natriuretic peptide. So atrial natriuretic peptide and brain natriuretic, what will be the signal for their release and where are they released?
Here in the atrium.
They are released in the atrium, right? In the heart, in the atrium, and also what the ventricle, right? Yeah, by the
Yes.
atrial and ventricular myocytes. So these guys, their signal is going to be volume overload, right? If there's
Yes.
increased volume, right? If there's volume overload, that will basically stretch, right? The heart, there will be increased blood to the heart, that will stretch the heart, and we're going to release what? We're going to release AMP, right? And we're going to release BNP. These guys, they basically counteract what? The effect of aldosterone. Remember, aldosterone and angiotensin, they increase sodium, right? Their major target is sodium reabsorption, right? Does that make sense?
Yeah.
And then water follows the sodium.
Yes.
AMP, BNP will inhibit, they counteract the effect of RAS, basically. So they short, basically, you can see them as counteracting the effect of RAS. So we're going to have what? Increased sodium, right? Excretion, right? In the
Okay.
urine. And then water too will follow, right? In that
Yeah.
case. So by doing this, they're trying to what? Bring the volume back down, right? To
Okay.
normal, the blood volume back to normal, right? So that's going to be our AMP, right? And BNP. You got
Okay.
this? Awesome. Good. So we've got a natriuretic, right? Peptide, right? This is right. Okay, good. So let's look at AD, and this is the reason, right? This is the reason why people with SIDH, right? Initially, you may be thinking that if somebody has SIDH, syndrome of inappropriate antidiuretic hormone, right? There should be volume overload. Remember, ADH primarily targets what? Water, right? It's pure water, not sodium. It's going to be water, free water. So you may be thinking initially that people with SIDH, right, they should be hypervolemic, right? Since they concentrate the urine so much, right? They
Okay.
are reabsorbing so much free water. You're going to be thinking, okay, should they be volume overloaded and whatnot? But actually, they are not. You get it? The reason is because if this somebody has SIDH, what happens to volume? Volume goes up, right?
Yeah.
Yeah, volume goes up. If volume goes up, what happens to sodium? Sodium becomes diluted, right?
Yes.
Yeah, so sodium concentration goes down, right? So if volume goes up initially, right, the volume gets to the heart. That will trigger what? AMP, BNP release. Makes sense?
Yeah.
And then that will counteract the effect of what? It's going to
Yeah.
inhibit, basically counteract the effect of aldosterone. And then we're going to have increased sodium excretion, right? And water will follow, right? So the initial water, the water that will be lost will be the same amount of water, right? That ADH, right? Increase its reabsorption into the blood. But now they're going to be losing more sodium. Remember, the water already diluted sodium, and now they are losing sodium again, right? So the patient volume will go back to normal, right? Blood volume go back to normal, but the serum sodium will still be what? It's going to be low. So that's why people with SIDA, they are euvolemic, right? They have euvolemic,
All right.
right? Hyponatremia.
All right.
Do you
So
get
I
it?
have a question regarding this. Like, if we have high sodium in the body,
Mm-hmm.
that means low sodium in the urine, right?
If the brain has high sodium in the blood, right, or in the body, if
Okay.
the kidneys are working well, their job will be to get rid of the excess sodium.
Okay.
Does that make
Because
sense?
in some questions I read like urinary sodium is high,
Mm-hmm.
and body in the body there is like more water and it's diluted, and urinary body sodium is high and urinary sodium is low. So is that how they compensate or they manage balance?
If what happens is no, in that kind of scenario there's a problem, right? Because if the kidneys are working well, if urinary sodium, if the body sodium is high, right, serum sodium is high,
Uh,
the kidney should compensate, right, and kind of get rid of excess sodium. So you should also see high urinary sodium, right,
okay.
until the serum sodium is corrected.
All right.
If the serum sodium is low, urine sodium too will be low, because remember, where are we getting urine sodium from? It's from the plasma sodium, right? When you
Yeah.
filter plasma, right?
Yes.
So, because if you look at your filtration is equals to what? GFR, right?
Okay.
Times what? Times plasma concentration. Do you get it? Like filtered load of any molecule? It's going to
Okay.
be the glomerular filtration rate times plasma concentration. So basically, filtration or the filtered load is what? Is a reflection of the plasma concentration.
Okay.
If I have high plasma concentration, I'm going to be filtering more sodium right into the
Okay.
renal tubule so that I can get rid of the excess. Do you
All
get
right.
it? If I have
Uh-huh.
low serum sodium, I'm going to be filtering less. less
Okay.
sodium into the what? Into the renal tubule because I have less plasma sodium. Does this make sense? Right.
Yes.
That's a normal for a normal functioning kidney.
All right.
But if a patient has hypernatremia or hyponatremia, right, is usually due to some sort of derangement, right? Something is wrong somewhere. Yeah.
Okay.
So in that case, you can see an abnormality, right, where the serum sodium can be low and the urine sodium is still high or something like that. Or you see serum sodium can be high, urine sodium is low, right? Vice versa, right? That is there's a pathology going on there, right? That is
Okay.
causing that abnormal, right,
Yeah.
problem where the kidneys cannot get rid of what the excess, right, sodium in the serum. Does this make
Okay.
sense? You
Yeah.
get,
Yeah. Yeah.
yeah. So
Now
that
I
would
get.
be the principle behind that. Good. So yeah, but this is the reason why patient with SIDH do we have euvolemic, right, hyponatremia, right? Yeah.
Okay.
But right, you got it. Good.
Yes.
When you went to hyponatremia, that was very clear, right, for you?
Mm-hmm. Mm-hmm.
Okay.
Yeah.
All right. Good. Okay. Good. 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 All right. So let's see. Okay. So we got right. All right. Now let's talk about ADH, right? Yeah.
Yeah.
So ADH, antidiuretic hormone. The
Yeah.
first thing, right? We're gonna first of all look at the key nephron, right? Without ADH, we want to see what will be the concentration of urine, right?
Mm-hmm.
Through each segment of the nephron, right? Without ADH, and then we're gonna come back and see what will happen, right, in the presence of ADH. Are you with
Okay.
me? You got
Yeah.
it? Good.
Yeah.
All right, so let's start. So first of all, let's start from the beginning. So we filter plasma, right? We're gonna filter plasma into the what? Into so we're starting from here. So we filter plasma here into the what? Into the Bowman space, right? Bowman space. The concentration of the ultrafiltrate here at the Bowman space will be the same as plasma concentration. Does that make sense?
Yes.
Yeah. Right. So this is gonna be what ISO, right? ISO osmotic, right?
Osmotic.
Right. So isoosmotic fluid. Good. In the what Bowman space. Good. When we get to so at the end, right? So now the fluid will go through the what? It's gonna go through the PCT, right? To the
Uh-huh.
PCT.
Yeah.
And the PCT ends here. So at the end of the PCT, right? What happens that what the fluid will still be is still gonna be what isoosmotic
Osmotic.
because of what you are reabsorbing the same concentration of sodium and water. Does that make sense?
Yes.
We are at the PCT, we do what called isoosmolar, right? Reabsorption of sodium and water, right? We are reabsorbing the same concentration of solute, sodium, and the same concentration of water. So the solution that will remain will still be what? It's going to be isoosmotic, right? Same concentration as what? As plasma. Are we on the
Yes.
same page? So
Yes.
far, so good? All right, good.
Yeah.
Now, right, good. Now, when we get to the what? When we get to the descending limb, right?
Okay.
Thin descending limb of Henle. This thin descending limb of Henle, right? It is what? It is permeable to water, but not to what? But not to solute.
Sodium.
So it is permeable to water, but not to any solute, right? Does that make sense? So
Yeah.
as urine is going through, right, this area, and it gets to the bottom, right, bottom of or the loop here, at the tip of this loop here, the urine becomes what? It's going to be hyperosmotic. Does this make sense, right? It's because we have a lot of solute remaining, right, in the what? In the tubule. We have less water now. We have a lot of water in the interstitium. We have a lot of solute in the tubule now. This is going to be what? Hyper, right?
Yes.
Osmotic. Going to
Okay.
be much more concentrated, right, than plasma. Awesome,
Okay. Yeah.
right? And then when you now go back up, by the time you get to the thick ascending limb, right, of Henle, you now have your what? Your sodium chloride, potassium, right, for transporter,
Yes.
right? And this guy is gonna be what? So this segment is permeable to what? Solute, right, but not to water. So now we're gonna be reabsorbing a lot of solute here. By the time you finish, by the time you go through this thick ascending limb of Henle, right, the urine will be what? Is gonna be diluted now. That's why they call this area the diluting segment. Yeah.
Okay.
So the urine is gonna be dilute. It's gonna be much more dilute than what? Than plasma. You get it?
Okay.
So
Yeah.
hypoosmotic, right? Solution good. Then by the time we go through these DCT, right? And DCT here, the major absorption will be what? Sodium and chloride. You get it?
Yes.
Sodium chloride, good. So now you are resolving more solute. So what happens? By the time we get to the end of the DCT, the urine reward is going to be much more dilute also. Do you get it? Like more
Yeah.
dilute than before we went to, right? So it's going to be also
Okay.
more hyposmotic because you are resolving more what? Yeah, solute again, right? Sodium
Solute.
chloride, right? And then we now get to the what? We get to the collecting tubule. At the collecting tubule, in response to at the collecting tubule here, in response to aldosterone, yeah, more sodium will be absorbed again, right? are going to be excreting potassium, but more sodium will be absorbed with the water. So the urine here should also what? Yeah, it's going to be diluted, right? More dilute. And then by the time you get to this end, it's going to be what? Very, very dilute. So the amount without ADH, right, the urine that will be excreted will be very, very dilute. Much more,
Okay.
it's going to contain a lot of water. We're going to be losing a lot of free water. That's why people that have what? That do not have ADH at all, or the kidneys are not responding to ADH, right? You have what? Polyuria, polydipsia, right? Excess free water loss, and that will lead to what? That will lead to hypernatremia, right? The sodium in the blood will be concentrated. Does this make sense? You get
Yeah.
it? All right, good. Now, let's switch to ADH now. So let's see what happens in the presence of ADH.
Okay.
So when ADH is available now, right? Something interesting happens. So what happens is ADH is gonna work like there are like two locations that ADH will work on. ADH will work on what? ADH is gonna work here. Give me a moment. So ADH works here. It's gonna work on this segment, right?
Okay.
It's gonna work. It has an effect on the thick ascending limb, right, of the loop of Henle, and then ADH works here. Awesome.
Yeah.
You get it? It's gonna work on the collecting duct or collecting tubule. All right. So let's look at let's break it down, right? What will ADH do? So we know that ADH has what? Antidiuretic hormone, or also called, which is also called vasopressin, right? It also has like two receptors, right? Correct? The first receptor is going to be what? The V1 receptor. V1 receptor is the GQ receptor, right? So it's going to work. So when we release, and remember, what will be the trigger for ADH release? Low volume, right?
Low volume. Yes.
Low volume or low what? Concentration, right? Osmolarity, right? That would be, sorry, high. If I lose volume, right, the osmolarity in the blood will be high, right? Yeah, like if I'm thirsty or dehydrated and stuff. Awesome, good. So that will trigger ADH, right? When I trigger ADH works, so it has a V1 receptor on the blood vessel, right, which is a GQ, right? GQ receptor, and that will cause what? Vasoconstriction, correct? Yeah, increased tone of the vessels. And then ADH works through V2 receptor, which will be in the kidneys now, right? Yeah.
Yes.
And this receptor is what? Is a GS receptor. Right, GS receptor. Good. So in response to V2 receptor, right? First thing, V2 receptor will stimulate ADH in the what's the name? In the thick ascending limb of Henle, right? Thick ascending limb of Henle is going to stimulate this transporter, sodium potassium chloride cotransporter. Okay. You got that? It's going to stimulate the sodium potassium chloride cotransporter. So what happens? We're going to be pumping more sodium, right? More solutes into the interstitium. Can you see what is happening? We're going to be pumping a lot of solute into the interstitium. When you're pumping solute into the interstitium, right? So we have a lot of solute in the interstitium here at this segment. What do you think will happen to the interstitial osmolarity? It's going to go up, right? The osmolarity in the interstitium becomes increased, becomes more concentrated. Does this make sense? The interstitial osmolarity becomes concentrated, right? Good. Now, if the osmolarity in the interstitium is concentrated, right, how much water do you think will be reabsorbed along the thin descending limb of Henle? A lot of water, right? Yeah, because that high interstitial osmolarity will start drawing water, right, from this segment. Do you get it? Yeah, from the thin descending limb of Henle, right, into the interstitium. So what happens to the urine that will be in the lumen? It's going to become much more concentrated by the time it gets to the tip, right, of the loop of Henle. Does this make sense?
Yeah.
You got it?
Yes.
Good. This is what we call countercurrent, right, multiplication. So what happens, right? This is countercurrent multiplication, right? So by increasing concentration of sodium, by stimulating sodium potassium chloride, you are increasing what? The concentration of the interstitium. This is countercurrent multiplication. You are adding more solute to the interstitium, increasing their concentration, right? And that will allow what? Movement of water out of the tubule. Any part of the tubule that is permeable to water, right, is going to what? Draw water, right? The interstitium will draw water out of the out of the urine or filtrate and make the urine much more concentrated. Does this make sense? You get it? Good. Then all right, so let's say we draw water in, right? We are drawing water. Another thing that will contribute to this interstitial osmolarity, right, or countercurrent multiplication, right, is gonna be urea. So in response to ADH, right, the cells of the collecting tubule, do you get it? Of the collecting tubule here, they're gonna start what? They're gonna put urea channel on their on their membrane. So they're gonna become permeable to urea. and
Okay.
urea will start what? Becoming secreted, right? Into the
Okay.
what? From the urine. We're not taking urea from the urine, we're going to be
Uh-huh.
secreting it into what? Into the interstitium. So more urea. So urea too is an osmotically active compound, right? It's going to
Okay.
contribute to the what? To the interstitial osmolarity. Awesome. Good.
Yeah. Yeah.
And then we
Yeah.
know that what finally what happens, we're going to put what? Aquaporins, right? In this
According to
tool. We're going to put our aquaporins, that's on the ADH. We're going to put aquaporins on the what? On the collecting tool. So what happens now? So you put aquaporins, the tubules are permeable now to water, and now you have created a concentration gradient that will draw water, right? Into the what? From the renal tubule into the what? Interstitium, right? So water will start moving
Okay.
right into this location. Does this make sense? You get
Okay.
it?
Yeah. Yeah.
Yeah. Are we on the same page?
Yeah. Yeah. Yes.
Good. Right. So that's what happens here. So this is what I'm doing. So what do you think will happen to the urine that will eventually get here? The urine that will get here will be very, very right
Concentrate. Yeah.
concentrated. Thank you. Because you're able to successfully reabsorb all the water. Yeah. So ADH increases what? Countercurrent multiplication. What that basically means is that we are pumping more solids right into the interstitium. Sodium, potassium,
Yeah.
ATP, chloride, co-transporter, urea, right? Cycle or whatever, right? We're going to be pumping urea into the interstitium. So that will create what? Interstitial osmolarity. And now we make the what? So every area of the tubule that is permeable to water, the thin descending lip of Henle, and also what the collecting ducts now in response to ADH will be permeable to water. They're gonna what? Start pumping water, right? Sorry, start reabsorbing water right into the interstitium. So the next thing
Yeah.
you have to do now, what? Get the water right from the
Yeah.
interstitium into what? Into the what? Into the blood vessel, right? Yeah. So that's where your vasa recta. Remember vasa rectors? Vasa rectors have what? Very, very long straight vessels, right? In the interstitium, in the
Yeah.
medulla of the kidneys. So the vasa
Yes.
recta, they're gonna be here. Basically, your efferent arteriole, right, is gonna
Yeah.
continue, right? It's gonna send in some vasa rectors, and right. So these are very, very long straight vessels in the interstitium, within the interstitium of the nephron or kidneys. So what happens? So they're gonna what? Because at this point now, remember what you did. When you get to the glomerulus, right, what do you remove from the blood vessel? You remove what? Water, right?
What?
Plasma. When we
Yeah.
filtered here, does that make
Yes.
sense?
Yes.
So what do you
Yes.
think will happen to the osmolarity in the what? In the blood vessel? The osmolarity is increased, right? They have a lot of solutes.
Yeah.
I don't know if that makes sense. So we
Yes.
have blood coming here. We remove the water, right?
Yeah.
At the glomerulus. We are going to be left with what? Solute, right?
Salutes.
In
Uh-huh.
the efferent arteriole. Those solids will go through the what? They're gonna go through the what? The vasa recta. They're gonna be the one remaining in the vasa recta, the straight
Okay.
vessels. Yeah. So those will create what? An osmotic gradient now that will now start drawing water, right? From the interstitium into the
Yeah.
what? Into the what? Into the blood vessels. Does that make sense? Yeah.
Yeah.
Water start moving into the blood vessels, and then the vasa recta will take the water, right, back into the blood, into back into circulation. Does this
Okay.
make sense? So this is
Yeah.
what we call countercurrent exchange now.
Uh-huh.
Yes. So
Yes.
countercurrent multiplication to increase the osmolarity in the interstitium, and then now we are taking the water right into the vasa recta to get into the
Okay.
blood vessels. Is
Okay.
this Does this principle make sense? You get
Yeah.
it? Good.
Yes.
So this right. So this is why ADH is important, right? So if somebody has so let's write out everything ADH do. So besides the vascular, we said that what also we're gonna what increase urea, right? Cycle, and then it makes the water permeable, the membrane permeable to what? Aquaporins, right? Yeah.
Yes.
So it'll be right. So the urine, final urine you're gonna get here is gonna be super, super
Concentrated.
concentrated.
Yes.
Imagine, right? Imagine this loop of Henle, right? The longer
Yeah.
the loop of Henle is, the more concentrated the urine will be, right? Because now
Yeah.
you have more chance for water to be reabsorbed. Does that make sense?
Yes.
Right. Also, the longer this collecting duct is, right, the more concentrated the urine will be. The longer the vasa recta is, right, the more concentrated the urine will be, right? Yeah, because we
Okay.
have more opportunity to reabsorb water. That
Water.
is why, yeah, the juxtaglomerular apparatus, right? Sorry, yeah, nephrons. Out of the nephrons, right, we have those nephrons that are in the cortex, right, and then we have those that are close to the medulla. You get it? The deeper you go in the medulla, right, the more concentrated the interstitium. So
Yeah.
it's going to be the juxtamedullary nephrons that will take part, right, in the concentration of the urine the most in this
Yeah.
countercurrent exchange, right, and multiplication. Does this make sense? You get
Yeah.
it? Yeah, good. And also animals like maybe if you compare humans to animals like camels, right?
Okay.
Camels that are desert animals, they have very, very long loop of Henle, very, very
Okay.
long vasa recta, right? so that they can concentrate their urine, right, and conserve water, right, in the desert. You get it? Yeah.
Oh,
So right. So that's the principle behind all this. Awesome. Good.
okay.
All
Yeah.
right. So right. So these are the iod hormones, right, that work on the kidney. I don't think did you miss any hormone? I don't think there's any other one. So prostaglandin and the RAS, ADH, BNP, ANP,